5 years ago, I was like you. I was here. I have seen some professors are present here. Hopefully, I will not do some stupid things and saying some stupid things. Today, I have another story because I spent like 10 years working by surfactants, and I moved between several years, labs, UK, Milan, Brescia. Then I said, like James said, it's the era of omics. So I decided to shift and to learn some, I would say, to to make this challenge and to go inside those tools. So today I have another story about cave microbiomes in the era of omics. I have two parts of this presentation. It's like to see an overview of cave microbiology research. James, he said there is several archives when we can deposit our sequences after sequencing coming from Illumina and Nobel and whatever. So we can have another few things if you work in the future about microbiome research, whether they get microbiome or whatever, you can understand what's going on in the world, what are the big labs and what they are doing. So, and then we end up like a model study in the cave microbiome in Rome. So you see this beautiful picture. This is my former PhD student, Paolo Torrini. He's like speleologist, and this is in New Guinea. It's another part near Terrestrial Australia, and he went to see how microbial community are able to adapt to those extreme environments. And he's fascinated with this, so he decided by the age of 50 to do a PhD. And I end up to help him during his third year. And we started to work another project. As you see his picture, it's like in the caves of Rome. We go underground below the city of Rome, and we understand what type of microbial community are able to adapt to those extreme environments. The last two pictures, this is like Emmanuel Charpentier caves. His name is Hazan from Icon University in UK. And she's really, if you are curious, you can Google her name and to understand about, she traveled all the world to understand about, to learn about microbial community in caves. So this microphone is something horrible, but I will try to do my best. I don't see the cursor anymore, but anyway. So by definition, if you think about biota of cave, we have 4 zones actually. Photic zone, we still have vegetation, microorganisms, and animals, and we have the twilight zone, microorganisms and animals, and we end up by the last we have the deep zone and the transition zone. So be careful when you go to cave from where you take your samples, and you need to well define your strategy of sampling. And then, as you see here, this is the gradient of the light. We still have from the light to the darkness. In the deep zone, we have more stable condition, and we have more I'd say, low biodiversity. Let me say, if we talk about biodiversity, this is people who think like this, but then with the sequencing technology, they discovered something different. Anyway, so as you know, all of you, beside the culture-based approach, we have new tools now in the market to make the ecology, and we are able to whether we sequence the omplicon or whether we do metagenomic sequencing. For the omplicons, for sure, we have certain markers like 16S, 18S, ITS, and whatever, depending on the domain of life that we want to track. Metagenomics approach, I will give you an workflow that we could monitor and to adopt whether read-based or assembly-based approach. So in the next slide, you know better than me, Mohammed, he told me make a presentation, but this is, you know better than me, you need to select the right primers. I know that, for example, for the gate microbiome, they use V3, V4 region. For me, for example, in caves, in ecology, I use V5, V6. So from the 16S to make my, to analyze my samples. So Mohammed said about omics, you know, after sequencing, we end up with this is workflow, and we have, this is for sure, we need to extract very well the DNA. Remember that we have oligotrophic environment. We need to do correctly the extraction of the DNA, and we have the right quantity from the biomass to have a good quality of the DNA. We make like PCR controls if we don't have any inhibition, and then we prepare the libraries for Illumina. So at the moment, to analyze the sequences briefly, I can tell you there is 2 main algorithms, Data2 and Shine2, and we end up like a count table where we have a number of sequences that we identified and how many times they are present. It's like the number of reads. Parallelly, we have this is taxonomy based on the, I would say, the database that we selected to analyze our sequences, RTP or whatever. And then this is your generation, my friends. You need to learn coding because in this field, you need to use R to do this is beautiful picture, beta diversity, alpha diversity, taxonomy, and whatever using R. So I tell all the time my student in this field, you don't have future at the moment, and you need to learn coding to be a good microbiologist, and you can compete at the high level. So regarding the metagenomics, I put some prices because I know there is a flexible budget. At the moment, I sequence one sample with 30 euro per sample. It's close 100 dinar, but I don't know the price here in Tunisia. In shotgun sequencing, what does mean shotgun sequencing? We sequence everything, the microbial community, and then we need to adopt two strategies, whether the read-based, which is the more easy, and you can find it in literature. And there is famous labs like Sigata lab in Italy. They do this as metafallen, and they do the read-based approach. So the read-based approach actually, we end up also with a count table, but here we have how many reads they will map against this database, whether for the taxonomy or to infer the metabolic pathways from those samples. And this is recently now, even the gate microbiome, they started to make the assembly-based approach. That means we need to assemble genomes and we need to construct mags to check their quality with this algorithm. I wrote several algorithms for the assembly mega spaces and whatever. I tell my student all the time, select the recent one and stick with it. And you are not at the level to benchmark all the tools. This is for other people, not for us as microbiologists. So, and then when we have good mags, we started to map them to understand the relative abundance in our shotgun samples, and we make like the depth from those samples. So again, we can do this is we do this as phylogenomic comparative genomics and whatever if we have good samples. Anyway, at the moment we have at the price 250 euros. It's around 500 dinar per samples. I don't know the case in Tunisia. I have 6 years abroad. So in the history of cave microbiology, people, they were interested to study caves, starting by the early studies like microscopy, cultural approach. And as you see here, with the discovery of 16S, we started isolating new species, characterization. And you see by 2010, the discovery of Illumina, we will see several publications. They started to be interested to study the cave microbiology. I put here 2022, it's the first metagenome assembled from the cave. It's not the case for the human gut microbiome because it's 12 years ago to tell you that there is a lot of things that we could then in caves. If you see even the number of publications starting from 2010, it started to increase the number of publications. But anyway, I tell you another story. That cave microbiology, it's not the gut microbiology. You need to have someone crazy to go cave and take some samples at the same time. I will tell you another story later. So by saying the sequence archive, we noted that most of the studied caves, they are limestone caves and SAS caves, sulfuric acid caves, and volcanic caves. There's difference between them. I will not go into the, I would say, the formation and whatever, but even the microbial community could change a lot between them, especially for the case of SAS caves. We did like meta-analysis. I invite you all to keep the track because you can do not experiment, but you can perform meta-analysis in microbiome, gut microbiome, or whatever, but you need certain skills to select your samples and to follow certain criteria to have the right meta-analysis that you could publish it in the future, especially if you want to write a proposal or whatever. So we end up like to compare 44 samples coming from 16 years, and we created this pattern as you see here, and we demonstrated that we have similar profiles and pattern between limestone caves and volcanic caves. For the SAS caves, we see some rare taxa, they are not present. Some there, they are not absent. Even within the protobacteria, we see there is variability, and this is Campylobacteriodota, they are present in sulfuric caves. This has helped you also to see an overview. Imagine the case we have a project of a gut microbiome, we need to compare between healthy people and people that have disease. So you will do something like this if you analyze your samples. Anyway, so even the matrix, we noted there is differences. There is similarity between the biofilm and the sediment, but if you sample the groundwater, maybe you expect to have certain microbial communities, as you see here. We propose it, and this is a review, you will see it maybe after 2 weeks, like basic and future recommendation if you think in the future to make like cave microbiology research, starting from in situ studies, like define the photic zone, install data loggers to record temperature, humidity, and whatever. Certain microscopy photography techniques, because it's not easy to take picture in caves, it's very dark. And you can do isolation shift, you can culture inside the cave. And there is in-lab studies like cell counting. It's very, very important here because you need to have good quantity of biomass to succeed the DNA extraction without inhibition. So to compare ourselves with the human gut microbiome, there is new ideas that you could perform in this sphere. And we discovered that metatranscriptomics, metaproteomics, metabolomics that not applied yet in caves. So we can go in certain caves in Tunisia here, and we publish, it's very easy to publish anyway. So I will give you this as idea. I come to Rome, they told me, okay, we have a project to study the urban caves of Rome. Why to this? Actually, to understand the risk. When we have a risk of exposure to pollution, maybe we expect, I would say, another reaction from microbial community. And we discovered even in Rome, they don't know how many caves they have. So I saw a lot of Tunisians, they complain about bureaucracy, but I cannot tell you about the bureaucracy in Italy to convince Italian to go below and beneath the earth. I challenged for 1 year to convince the region of Lazio to go to this is 5 caves. I will show you this is caves. Anyway, we selected 5 caves. There is history for each cave. Like for Lanini, it was ex-hospital. Monteverdi, the first time after 30 years, we were me and Paolo Torrini to go there to study the microbial community from Monteverdi cavity. And we did certain controls, and this is Rome. Anyway, this beautiful picture, me and Paolo Torrini, we went the first time there to sample the groundwater and the sediment and the biofilm. All of them, they contain groundwater, and we need to see the shared taxa between the groundwater and the sediment and the biofilm. This is the workflow. So for the student, Mohammed told me to inspire young, you need to design your workflow from the beginning if you want to do omics studies. We have 5 caves. We sampled biofilm sediment, 2 replicates, and then 1 groundwater. We filtrated 2 liter to 4 liter. And then we amplified V5, V6. We end up with 25 FastQ files, and we end up with 4,225 ACVs. If I say ACVs, it's amplicon single variant. It's like operation taxonomic unit in the past. So, and you see this is a high number of ACVs, which indicates that we have higher biodiversity at those contaminated sites, at those caves. We did also physiochemical characterization. So do not get lost in omics without confirming with certain experiment. You do only omics, you get lost with a lot of idea, and then you are not sure with those ideas. What does it mean, this? If you have a project about omics, you need also parallelly to run certain controls to confirm your data. So we confirmed that we have oligotrophic environment at those sites. We excluded that we have contamination with hydrocarbons, and we have a higher contamination with higher concentration of phosphorus at those sites. So the microbial community, as you see in this graph, it's mainly colonized by protobacteria and Nitrospira. For the groundwater, we see something variant, different, I would say, at certain sites like Fiume, Epigeo, and Monteverdi. So here you remember when you do meta-taxonomic profiling, you need to profile your results at what taxonomic level? Phylum level, genus level, species level. If you do Illumina and Amplicon, you are unable to do the species level. So you must go to shotgun sequencing to profile your results at the species level. So keep in mind this. All these plots, they are lacking. There are taxa, you will not see them in your resolution of your results. Anyway, so even at the genus level, which is the maximum that we could obtain with Illumina sequencing. We have certain bugs like Chloromonas and Hydrogenophaga. We usually not find them in caves in general, in natural caves. And we found them in caves in Rome. This means we have a contamination in the past, despite that we have higher biodiversity, we don't know at the moment. And this is why we did another sampling campaign, and we're analyzing sequences to confirm those results. So even here, the index of biodiversity are very high for the biofilm and the sediment, lower for the, which is normal for the groundwater. And here this is like a Venn diagram analysis. We confirmed there is a lack of core microbiome. It's the shared taxa between those five caves. And this is shared taxa between 5 caves. It's easy, I would say, more easy to find it when you work in natural caves. But if you work in anthropized caves, maybe you will end up difficult and challenging to find this shared taxa. Can I ask you a question? How I obtained those metabolic pathways, especially I amplified only amplicons. So I don't have all the genomes. Could someone reply to me? Yes, there is database now, especially they are more developed in the human microbiome. You can put the amplicon sequences and you can base it on the taxonomy because you will have a lot of unclassified taxa. You can infer the metabolic pathway. But if you want an accurate resolution of your result, you must go to shotgun sequencing and you need to spend more money. That's it. Anyway, we confirm it in those sites. We have higher, the microbial community that are able to degrade hydrocarbon, they are mostly present. There is variability between them, but I will not go into those details. But anyway, problem, Mohammed? Sorry.
You noted something. My friend Alif, after all these years passed in Italy, when he talks, you noted that, no? He used Italian way.
The Italian, not only it's like the Mediterranean way, not only us. So how do you think? Anyway, I will not go key findings about this. We have high richness, proteobacteria mostly present. We have community assay, they are biomarker in those caves and potential to degrade hydrocarbon. This is very, okay, it's not side results, but the most important for me now today that I see this beautiful caves in Tunisia, they are not studied yet. I control it in all the archive and there is no submitted sequences. This is the future and your mission maybe will be crazy in the future to go inside those caves, especially that we have, this is Ain al-Dhab cave. I met several people from Europe, they have a wish to go to this because they have them, it's very beautiful to be honest, and they want to study it. But anyway, and even I was snaking I would say Facebook yesterday, I realized that in Tunisia, we have like next sake in Institute Pasteur, if I'm wrong, and we have my sake in Zeus, and I don't know the reality of this infrastructure here, especially in the public. So it's the future for the young generation to make this data and to submit it. So in this field, you have two options. You can publish even in Nature, and you can in junk journal, and you will happy with your results because they will trust you. And finally, I will tell you that for the young generation, there is like the new technology of hi-fi reads. Even the gut microbiome people, they were interested to apply them. My friend Martina, she's trying now to extract the DNA from in situ from those caves, and even RNA is difficult. But she's really crazy. This lady from Italy. There is a lot of idea that you can apply them. The most important thing, learn coding, please. You will see this review. You can, about the cave microbiology in general, we have our, I would say, guidelines. I come from Professor Paulo Visca. I have a history in this field. And we wrote this review about the cave microbiology research and the new ideas that we could perform them. Finally, I'm so grateful here. I thank my team. This is Professor Paulo Visca, and I will not name them all. And the people, they give us grants, and they were so crazy to work on caves. And thank you very much.
Merci, Elif. Thank you very much. So the floor is yours. Young people to ask him crazy questions about his crazy adventure in Rome caves.
No questions?
Yes. Vas-y.
Thank you very much.
You are inspiring our students. My question is, what technique are you using for each one? And
I saw
second one, what do you suggest for the biochemistry and how to conclude
Thank you very much. This is my professor. I'm happy that he's asking me this. He taught me in SBM. I was student in his class. I bought this class. So what I think in situ, I saw this is, she bought like mean island and she's trying. But you know that we have lower quality when we have long reads. And she's trying to make some analysis in situ. And also she's trying to reproduce the results in the lab. And she told me that she's drawn co-assembly, that she's long reads from the same samples and short reads, and then she co-assemble them together to have higher resolution. You know, it's the field. It's if you are rich, you can have the idea to do everything. But anyway, with sometime with the good idea, with one sample, you can publish in Nature. This field. So you can spend I don't know, 1,000 dinar and you have a good publication. And sometimes you end up with 16 shotgun samples and there is no controls, there is no, people that will not understand what you are doing. That's it. Hopefully I replied to your questions. My recommendation, co-assembly, and shotgun sequence. But I know it's expensive. But we can, in caves in Tunisia, we can start with Illumina.
From scratch.
Yes, we can start with Illumina, with MySec, and then we have metataxonomic profiling, whatever. And then we can, in the future, if you want to have certain part of this cave to analyze them. But remember, here you need to have, us as microbiologists, we cannot navigate caves. You need to have expert in biology. And you need to adhere with them, they will help you, and they will sell you if you do some stupid things there. That's it.
Any other question? So let me then ask you,
Okay, go ahead.
pour rebondir, deux questions.
In French, whatever. Okay.
Fundamental question. Was there any correlation
All right.
between all the cases you checked
The correlation is hard. What do you mean?
in terms of nature of bacteria?
Yes.
Because you know that I'm not in that field of bacteria. So, and second question is for rebondir, is there any advice to encourage the collaboration between scientists,
Sure.
archaeologists,
Yeah, that's what I.
geologists, you know, all these researchers and
So the correlation at higher taxonomic level, all of them, they are oligotrophic environment. So you will have end up with certain bacteria are able to thrive at those conditions. At higher taxonomic level, it's okay. But if you go down, maybe you will end up, you will not see common, let's say common taxa. But this is not the case for limestone caves. If you work in limestone caves, because the type of rock is not the same. Less than the volcanic caves, the type of rock is not the same. So you will, the bacteria that are able to colonize those rocks, they will be the same mostly, especially if you take them from the same zone, as I showed you there is first. The second question about collaboration, right? Yes, we can collaborate. Yes, I found a lot of people, even in Europe, they are stuck to analyze their sequences. So as a young, maybe master student, he could able to analyze this as FASTQ files. You will help him and you will get cited in his publication.
I have curiosity now.
Yes.
I look at James and I'm taking that.
Yes.
Do you think that this archaea bacteria in cave have a particular set of plasmid?
This is a good question.
No.
This is a good question. To be honest, with this is techniques of most of the techniques that we found them, it's most focalized in bacteria. But to identify archaea, you need to use the right primers to identify them. But I tell you, I saw some studies, they do shotgun sequencing, and they use certain database to infer how many drug-resistant genes. Those drug-resistant genes that could be in the plasmid part. So I saw some traits that there is something interesting. But remember, in my field, it's not the case in the gut microbiome. The gut microbiome is more robust field, and there is a lot of results and so on.
Yeah.
And that's it.
Yeah. Good, good. Any other question from the audience? Yes, go ahead.
Instead of figure out that
Exactly.
the reported in case of evidence of the other substance that bacteria can use for source of carbon.
Exactly. For my case now, we try to search and we look at 1,700 publications this PhD student now is working, and we try to classify all the family of pollutants that we could find them in tapes. Microplastics, heavy metals, hydrocarbons, and whatever. And then we can see how we can infer those metabolic pathways based on the activity of bacteria. But to be honest, I tell you from now, you need to go and do some experiment in the lab and to confirm that this bacteria is able to degrade this compound or whatever. All the omics, okay, you can infer and whatever, but it's based on the DNA.
Don't capture the don't make
This is the luck.
Yeah.
When you have DNA, otherwise you need to spend money and you extract the RNA and you merge all the results, RNA and DNA, and you see the expression of the genes. I saw it's more developed in the human microbiome. They have certain disease, they extract RNA and they map the reads of the RNA, cDNA, if you want. They map the reads after sequencing to the shutdown sequence. And they understand the metabolic pathways, the genes, not the protein. Hopefully I answered the question. Thank you very much.
D'autres questions? So, a big applause to my friend. Thank you very much.